Okay, so as in the last video, we have discussed about the introduction to cellular respiration, and we have discussed a bit about glycolysis. So let me give you the last video what we have discussed in the glycolysis. So we have basically break down this glucose molecule into two pyruvates. Now to break this glucose molecule into two pyruvates, the 10 steps are required. 10 steps. So today we're gonna discuss about those 10 steps. So those 10 steps are basically divided into two, we can say, steps, two forms. Number one is preparatory form. Preparatory. Another we can say oxidative. Okay, one is preparatory, another is oxidative. Now you might be thinking where this glycolysis process occurs. So this occurs in cytoplasm, either in bacteria, in we can say bacteria, fungi. This process occur in cytoplasm. In every we can say organism, cytoplasm. In animal, in plant, in bacteria, so this occur in cytoplasm. So in the preparatory phase we have the five steps, one further five steps we have in the we can say oxidative phase. Here we have furthermore five steps. Fine, so here these were discovered by three German scientists. So let me tell you the names of those three German scientists. Sorry, three German scientists. Number one is Embenden. Just remember these names because in the mostly testing these are to be asked. Embenden. Second we have Microf. Well, third we have Parnaz. Okay, so let's discuss this preparatory phase in further more detail. Fine. So here we are going with preparatory phase. Yes. Pre- So this preparatory phase is also known as investment phase. This preparatory phase is also known as investment phase because in the first five steps we are investing. We are giving ATP investing, okay? Investment phase. Okay. After that here we have what? In this how many ATP are consumed? So two molecules of ATP are consumed. No ATP is formed, only two are consumed. Fine. After that, here glucose molecule is basically break into how many? This is break into the end product of this preparatory phase is two molecules of G3P classic triphosphate. Okay, so now let's discuss these five steps. Okay, so number one, first phase is basically here we have first phase in which first of all glucose is break down into. Okay, in the first phase, for example, for suppose you can learn like this, we have one box, any box, and we have to add the straw, और हमें इसके अंदर straw add करनी है. So in the glucose, first of all, we will add the phosphate, equal to phosphate. Okay, we will add the phosphate. This straw is basically the phosphate. We will add the phosphate, and to add this phosphate, this will convert into 1,3. We can say this will convert it into basically glucose 6 phosphate. Glucose 6 phosphate. Okay, 6 molecules of glucose and we have add one phosphate. So how this phosphate come from? Okay, अभी phosphate आया कहाँ से? So yeah, here we have ATP break down into convert into ADP. ATP convert हो गई ADP के अंदर and we got one phosphate और हमें एक phosphate मिल गया. So this is done by enzyme that is hexokinase. Done by the enzyme that is hexokinase. Hexokinase. Fine. After that, I hope till the first one process is clear to you guys. In the next, we have another. Sorry. Yes. Another step we have basically second step in which this glucose 6 phosphate will convert into its we can say isomer in its own isomer glucose 6 phosphate will convert into isomer of this, that is fructose. Fructose is the isomer of glucose, so that is fructose 6 phosphate. Okay, glucose 6 phosphate. And here no ATP is required, only this is done by the enzyme, that is isomerase. 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 Okay. After that, in the third one step, we have in the third step, what we have, like we can say yes. Now from this fructose 6 phosphate, we have to break this. We have to break this. Matlab, here we were having this fructose 6 phosphate. So what has been formed here? For suppose the fruit was formed. Fruit was formed. Now this fruit, we have to break this fruit. So we'll add one phosphate was added here, and we'll add another phosphate here. Okay, one phosphate was added here. So here we will add another phosphate to break this fruit. Fine. So here we have. So how we gonna do this? We gonna convert this like yeah, fructose. It will be 1,6 bisphosphate. 1,6 bisphosphate, because as on the one we can say carbon, the phosphate will attach and we have attached another phosphate. So bis means 2. So 1,6 bisphosphate. Okay, another phosphate is being attached. Now here the enzyme is used, that is basically can say sorry, here no any enzyme is used, or I can't remember the name of that enzyme. After that, this 1,6 bisphosphate is converted into two steps, because this is very unstable, so it is converted into triose, three triose. Another we have three triose. This is converted into triose. So as you can see in the chemistry we have studied, these are converted into, we can say, one is ketone and one is aldehyde and ketone group. So here this will also convert into aldehyde, and this is converted into ketone. So it will become, we can say, glycerol. So it will become glycerol aldehyde, glycerol aldehyde 3 phosphate. Fine. These are basically two different forms, and this will become dihydroxyl acetone acetone phosphate. Fine. So now here we have what? Here we have three carbons here, and here we have also three carbons. Fine. So now this is the fourth step is done here. After that, in the fifth step, we have what? We have the splitting. Fifth step is basically named as no. Fifth step is named addition. So this So this dihydroxyl ketone will directly change into glyceraldehyde. Okay, ये क्या होगा? Change हो जाएगा glyceraldehyde के अंदर. So this is basically the reversible process. We can say reversible step which can be changeable into each other. So dihydroxyl acetone phosphate will directly convert it into glyceraldehyde. So now further the process will happen. So we will count the two molecules of glycerol अल्डिहाइड, क्योंकि ये भी हमारे पास कन्वर्ट हो गया ग्लाइसरॉल्डिहाइड के अंदर. सो नाउ वी विल काउंट टू molecules of aldehyde. So till that this is done. In the next video we're gonna discuss about oxidative phase, in which we will start from two glyceraldehyde, sorry, glyceraldehyde, not glyceric acid, two molecules of glyceric acid. There's two molecule of glyceric, two molecules of glyceric acid. Okay. Fine. So now here we're gonna write the equation what totally happened. So let me write here the equation full. Okay, here I'm going to write the equation. So first of all, what G3P was formed? Sorry, glucose, not G3P. I'm going to use the glucose. In the reactant, we were having the glucose, which glucose was break by the help of 2 ATP. We used 2 ATP, and it formed what we got G3P gastric acid 3 phosphate, plus we got 2 molecules of ADP. ADP. I hope till that it is clear to you guys. So in the next video we will not go further from here, because now here in the next video we will count further from 2 molecules of G gastric acid aldehyde. Bye bye.